Hello, College Biology. This is Nucleic Acids Part 3. We're going to talk about the polymers of nucleic acids. There are two primary polymers of nucleic acids. Those are RNA and DNA. RNA is ribonucleic acid. DNA is deoxyribonucleic acid. And they are determined by the sugar that is in those nucleotides. If it is deoxyribose in the nucleotides, those nucleotides will make DNA. And if it is ribose in the nucleotides, those nucleotides will make RNA. So we'll start with the shape. DNA is famous for its double helix shape. It's made of two strands, and the nitrogenous bases form hydrogen bonds with each other, which unite them and form this helix shape, whereas RNA is a single strand. Now, they have it in a helix shape here, but honestly, that's not always the case. It's really more stringy and floppy, and it can float around, and sometimes it curls over and makes hydrogen bonds with itself and gets itself kind of in a knottish type. It's kind of a wild string shape. So RNA is far less organized than this helix shape suggests. So this is a little bit of a misrepresentation of RNA. It's definitely not this ordered. It's not like this at all. It's just kind of a goofy strand. So that's one difference between them. Another difference between them is their function and their length. DNA is 100 to 200 million nucleotides long. It's a very long molecule, and that is and we have many strands of DNA, but amongst those many strands hold our 20,000 genes, whereas RNA is only about 300 to 150 nucleotides long, and that would be the length of one gene, and that's because DNA contains all the genes of an organism, and RNA is a copy of one of those genes. The reason why we can use that term, genes, this way is because the term gene means instructions for making protein. So, the instructions for all of the protein of the organisms are contained in DNA, and each RNA strand, each mRNA strand, is the instructions for one protein. So, it's going to be much shorter than a strand of DNA. Let's take another look at this. DNA, this double helix molecule that contains all of the genes of an organism, is contained in the nucleus. We keep it there. If you're a eukaryote, if you have a nucleus, you keep the DNA in the nucleus to keep that DNA safe. Well, RNA is copied in the nucleus, but then it is sent out of the nucleus to a ribosome, which actually reads it in order to put together a protein. So that's another difference between them. Furthermore, DNA, the nitrogenous bases of DNA, determine what the nitrogenous bases in the RNA are going to be. And the nitrogenous bases in the RNA are going to determine what monomers the ribosome uses to make the protein. So there it is. Those are the differences. Let's review. The polymers of nucleic acids are DNA and RNA. DNA is deoxyribonucleic acid. It has a double helix shape. It contains all of the genes for an organism, and a gene is the instruction to make one protein. And human cells have 20,000 genes per human cell, and all of those cells have the same genes as each other. And DNA is found in the nucleus, and the order of nitrogenous bases in DNA determines the nitrogenous bases of RNA. Where RNA is a single strand, it is a copy of one gene. It is copied from DNA in the nucleus, but is sent out of the nucleus to go to a ribosome, and the order of nitrogenous bases in RNA determines the monomers of the protein. We will talk more about chromosomes, which are sort of modified strands of DNA, in the next video. I hope this helps. Struggle well.